Okay. So, hi, I'm Chengwen, and I'm from the Institute of Biophysics, Chinese Academy of Science. So, my poster title of this study is the 70 CBV fMRI reveal the cortical microcircuit of bottom-up saliency in the human brain. And we can see that a visual atom in sharp contrast with its neighboring atoms in a simple feature such as color and orientation automatically capture attention. This is called the salience attention or external attention. And the possible pathway for saliency signals, it have two different possible pathways. One is that the saliency signals is created in V1 and feed forward to the higher order area, such as intraparietal circles, and an alternative possible pathway is that the saliency map of attention initially imaged in the frontal parietal area and then modulate the early visual activity through the feedback connections. So to reveal the cortical microcircuits and bottom-up saliency in the human brain, we investigate the cortical layer dependent CBV activity to orientation defined saliency stimuli in early visual and parietal cortex at 70. So this is the stimuli and procedure for the saliency experiment. And the orientation contrast was present at 0 and 15 and 19 orientation contrast degree. And these conditions were presented in separate 13 seconds blocks, and one stimulus blocks consists of 16 bar display, and each lasting 200 milliseconds and followed by 200 to 400 milliseconds of fixation. And the foreground bars will appear laterally to the left or the right set of the fixation. And for the retinal topic localizer, the colored natural images were present as the corresponding foreground locations of the saliency experiments, and the functional data of CBV were collected with a vessel sequence. So the sequence parameters were mostly from the length. So and the T1 anatomic value was acquired with MP2 rich sequence, and this is the activation map from the localizer stimuli, and the red and blue circles indicate the ROI for the foreground and the background regions, and then we got the layer dependent response in the foreground ROS in finger we also get the response, layer response difference between the foreground region and the background bars. And in finger A and finger C, we can see the normalized CBV response to the three orientation contrast conditions. And in all cortical areas, we can also see that the higher orientation contrast have the stronger fMRI response. And in finger B and in finger D, to minimize the difference of the top-down priority signals, we calculate the saliency response as the response difference between the 15 and 19 degree orientation contrast conditions. in these two results thinker, we can is show that in V1 the saliency signal was stronger in superficial depths, but and in V2-3 and IPS the saliency signal was peaked in middle depths. This is all same in foreground ROS or and the response difference between the foreground region and the background bars. Except the fMRI experiment, we also perform a behavior experiment to measure the subject's contrast detection performance. And we can see in this figure the contrast sensitivity of the 90 degree was higher and then 15 degree. And we then calculate the correlation between the orientation saliency response and the contrast detection performance difference across the depth of the V1. So we can say that the superficial depth in V1 the correlation is greater than the deep and middle layer. So we can this is our conclusions. First is that CBV response from the orientation saliency was the strongest in the superficial depths of V1 and peaked in the middle cortical depths of V2-3 and IPS. And then the saliency response in the superficial depths of V1 was correlated with the contrast detection performance. And the next is the bottom-up saliency map is initially created by the SO feature expression, so the natural inhabitation in V1 superficial layer, and then projecting to the higher order parity area IPS through the feedback pathway to generate the attention priority map. So this is the possible pathway for the bottom-up saliency singulars. The spot hypothesis. So this is a study